No, no, it's up to you. No, no, do go for it. Yes.
I request the chairpersons to please take over the session.
Good morning. So today's lead talk will be given by Dr. Subeer Majumdar. So I know Subeer when he was in National Institute of Immunology long time ago, and our researches coincided or overlapped on sperm physiology. Since then, he has moved to several other places where he is the director. So he has lots of publications, but unfortunately, I have not kept up with these areas after he left the sperm physiology. But looks like, based on his abstract, he still has one foot in male fertility and testis, while he has been pursuing, newer areas of research. So his talk would be on, let me see if I can, yeah. So exploring cells for innovative technologies. So Subeer Majumdar, please.
Thank you very much. So, good morning, friends, and welcome to this session. Good morning, chairpersons. Thank you for inviting me. Thanks to Professor Algur, Vice Chancellor of this prestigious university, and my other friends and colleagues and students who are here. And I will not waste time, and I will try to finish in time. So 45 minutes, I will try to finish in 40 minutes so that we can speak for 5 minutes. So I will be talking about, ये होता नहीं क्या? Okay. So Professor Algur, I will try to see you are an engineer, and my job is to ensure that what I say, at least 50% of that you understand. And that is what students should learn, that how to make presentation, that everybody sitting in the audience must learn. So if you see through first few slides, these are all our normal life things, what I'm going to talk. And exploiting cells for innovative technologies, it doesn't work on this, isn't it? Huh? It doesn't work. That's fine. Huh? Fine. That's fine. Okay, I can do that. So these are my students, and this is our institutes where I worked. And the first thing I will tell, that market for technology development and commercialization of the cell-based diagnostic test for cancer management, hematology, histopathology, microbiology, blood product safety, tissue transplant management, prenatal testing, this is all lying in front of you, which is all based on cell-based technology. Now I will go to the origin of the cell therapy, can perhaps be traced. You can see Charles Edward Brown Squared in 1870 to 94, he injected animal testicle extract in an attempt to stop the effect of the aging. So we all are worried about aging. So he did that, and that is the first, one would say, cell-based therapy in the world. Now today's day, we are seeing CAR T cell therapy and several other T cell therapy, where blood from the human beings are taken out, and blood cells are either given the genes or exposed to genes so that they respond and becomes a anti-cancer responsive cell, or near the cancer tissue, if there are blood cells, that can be taken and then that can be multiplied 1,000-folds and given back to human to fight cancer. So this is another way. What is cell technology? At its core, cell technology involves tools and techniques designed to study and manipulate cells, the basic building blocks of life. It includes methods for isolating, analyzing, and doing a lot of molecular biology to understand the cell. In blue color it is written, it enables scientists and clinicians to observe cellular process in real time, identify disease markers and develop targeted therapies. I will not go much because time is less. Now somatic cell therapy are of two kinds. In somatic gene therapy, the somatic cells of a patient are targeted for foreign gene transfer. That means my liver or brain, if has a problem, can be directly manipulated while they are inside my body, or they can be manipulated outside and given back to me. And germline therapy, that means if you do it in testis or ovary, every progeny will not suffer from that disease. It's completely wiped out. Now basically, if you do the targeted gene mutations, so you see, for example, there is a person who has some mutation and he is suffering from a disease, you can do targeted gene insertion to remove this bad part of the DNA and insert the good part so that it can be given back to human and that man will remain good forever. So he will be out of the disease. Now how the cell technology works, I will skip this slide because one most important thing is genetic and molecular profiling of cells, that means DNA, microRNA, so many other things, transcriptomics, proteomics, and manipulation and genetic engineering. Once you know the knowledge about genes, protein, transcription factor, you can manipulate them and try to make human beings good. Today, at the bottom, we say data interpretation. So advanced software and artificial intelligence analysis of various vast data sets to identify patterns, biomarkers, or therapeutic targets. Now, cell-based innovations are very, very important, but I would like to say at today's date, many transgenic animals are made using the cells, manipulation of the cells, so that we have a disease model. There are CAR T cell therapy, we know that. There are T cell reprogramming, there are IPS, that is induced pluripotent cells. We can create artificial organs. Promoters of specific to a cell type have helped. That means if you liver has a few genes which are secreted, they have specific promoters. All our body has same chromosome, brain, liver, kidney. But kisi ka chadar somebody's, what you call as bed sheet is open, so that gene will transcribe in brain. Some other bed sheet is open in my liver, so that gene will transcribe in liver. So remember, that's very, very important. And I will tell you, for students, mainly I saw a lot of students, so I made this talk more as a student prone talk. Now, if you see that several technologies, next generation sequencing, proteomics, metabolics, biomics, have revolutionized biological sciences and medicine. Blue, with modern tools of the bioinformatics, researchers are able to assign functions to several of such identified genes, whether plant science, whether animal science, or any science, or bacteria, or microbes. When you know about the genes, you have to know about its function. Now that is very, very important. For knowing the function, you have to make a whole live organism or a plant and then see how it helps. So what the machine gives us is only the sequencing, and they will say, I have found 3,944 genes in this species. We don't know what is the function of each gene. For that, what we need, so they are for us like these medals of a general or admiral. Only he knows what it is. We don't know what are the different symbols mean. So we have to cut each and every gene and try to put it in a plant or an animal and try to the function of it. That is called as transgenetics. In human beings, or for animals, we can create disease models manipulating genes. And actually, Lothrop and Loeb published the effects of hormones on development tumors in mice in 1916. So, very good mice models came up from them and a lot of work happened, organized. But now, certain diseases that affect humans but normally do not strike mice, that means you cannot have Alzheimer's mice, you cannot have a cystic fibrosis mice, but you can insert genes and now convert that mice like a human, giving cystic fibrosis in lungs or giving Alzheimer's in the brain. So you have to make that model of animal so that you have a human model. So that's what is called as transgenesis. This is a growth hormone human being, overexpressing. There is an American lady, must be around 6 feet tall, but look at him, how long he is. Can I do experiment with him? I cannot. They will not allow me to do experiment with human. So what I have to do technically is I have to, first mouse created was bovine growth hormone gene overexpression mice or rat. You will see that their body weight is at least 1.8 fold more to 2-fold more. Length is more. So now I have a model. This man is only alone, and in whole world I can find, let us say, 290 such overtly expressed growth hormone person, but I can have thousands of such mice to study progression of disease, to study how to counteract the bad effects of the disease. So transgenic mice are important. Now I'll go for the students. So when we see chromosomes, what we see that it will unwind and we will see two things. One is promoter. So machine reads and says, yahan se the gene starts from here and there's a termination codon. The machine I have shown you gives you only that. Now I'll say, okay, what does gene number 3940 does? No one knows. So you can cut this gene by DNA cutting technologies called as genetic engineering and paste it into the genome of mice. And let us say the mice becomes bald. So you know that gene was important for hair. Let us say mice becomes suffering from loose muscles, can't walk properly. So you know that gene was important for muscle strength. So you have to cut and paste, and that is called as genetic engineering. So you have to remember that's what is a gene, promoter and termination codon. These are the people for students, just to get excited and get little change, don't sleep, who gave you the structure of DNA, and now we can work with that. So what I was saying, in a cell, there is a promoter engine which drives a gene. This is the actual per se thing which is going to transcription, translation. Only pipe is important for function. This engine is cell specific. Liver mein liver ka engine hai, brain ka engine hai, right? But what is important is protein. So when this promoter drives, its protein in the brain is secreted. We call it glial derived neurotrophic factor, GDNF. This is termination codon. Now you can do one wonderful thing in transgenesis. You can keep the promoter there, cut this and throw this away and put human gene of disease in it and express that in the brain if it is a brain promoter. So you will have Alzheimer's or any other disease related to brain. This is beauty of transgenesis. You can do a lot of cut pasting, engineering of the genes. We call it genetic engineering. So what we know, when the machine gives you the bogey, the termination codon, everything, we have exons, we have introns. What we do not know where the train is going. What is the function? What is the function of this train? So that is why we are there to make transgenic animals and try to find out what these genes does. How to insert this gene into mice genome? So during cell division, lot of DNA भांगड़ा देखा है कभी? सरदार जी लोगों की शादी होती है तो दिल्ली में कौन क्या कर रहा है, क्यों नाच रहा है, कहाँ नाच रहा है, पता नहीं लगता है. Everybody is mixed up. This is the way embryo में cell divide होता है. माँ के पेट में अगर आप माइस का फुल बच्चा 21 days it is ready. You can imagine lungs, liver, everything is ready in 21 days. At that bhangra, when cells is going haywire, if you throw your genes, that's what we do, it gets integrated. And that's how human genes can be put in animals. First animal made in the country was at National Institute of Immunology, the lab which I led. But before me, the person who led made this mice, not me. And that is the HBVX gene, hepatitis B. We said hepatitis cancer hoga. So they isolated that gene, HBVX, hepatitis VX gene, and inserted in that mice, as I said, DNA. And the mice transgenic born had a very big liver. Compared to its brother of the same age, was very small liver. So we have now a model of hepatitis-based cancer. You can have hundreds of such animals and test any drug you want, which you cannot test in human. But you have a model of hepatitis cancer. So this is a cell, egg. This is the cell. I am again talking about cell-based technology. And in this cell, we insert a foreign gene. And that's the red one is foreign gene, maroon one is native gene. And this red gene will go and put its head and sit there on that maroon gene. That is a transgene. And that's called as, process is called as transgenesis. I'll give you a very good example. So this is a polio virus. So when polio virus tries to infect a human, it infects because this is a receptor of polio virus. It goes inside the cell of human and there is a poliomyelitis, leg has problem to walk. If you go to a mice, there is no receptor. You give thousands of polio injections, people did, oh, mice first class is to run. Nothing happened to mice. So what they did, genetic engineering, they cut this gene from human genome and made a transgenic mice and inserted this. As soon as they did that, the virus came there, sat there, and it has all properties of the poliomyelitis. So you had a wonderful model where you can study poliomyelitis. So you have to remember, this is a micro manipulation assembly where such kind of transgenic work is done. This is the egg, single cell, where foreign gene is inserted, small pieces of foreign genes are inserted, and then they are allowed to become embryo. Then from that one cell, since we have inserted the gene, every cell which divides gets that. If you are lucky, you can generate transgenic animals from that. Iske beech mein click kare. Please click in the center. So this is an egg, and this is how in egg, we routinely used to do that in National Institute of Immunology. This is that foreign gene of poliomyelitis receptor or brain, Alzheimer's, some genes. We'll insert it in the egg. And this is male pronucleus, this is female pronucleus. Male pronucleus is a little bigger. We'll throw it here, hundreds of eggs are lying, and technical officer will constantly take hundreds of eggs, inject the gene, go further. This is what is a very routine phenomenon. Other egg will be caught from there and brought. You can see one more sperm is trying to enter, is moving. But it cannot enter because after one, no sperm is allowed to enter. So this is what we do and generate transgenic animals. It's not easy. Now I will come. This is a society for molecular signaling. So from cell technology, now I will come using cell technology, addressing molecular signaling. Sir, when it is 40 minutes, can you tell me? Whenever it is 40 minutes. So I will finish in 2 more minutes. Somebody is keeping time? I'll stop wherever I am, doesn't matter, as long as you are enjoying. Are you enjoying? Very good. So Indrashis Bhattacharya, the local man here, and myself were having I used to walk from 9:45 in the night to 10:30, and one student will walk with me, so Indrashis Ganguly, and I would use that time for talking. So we were talking that there is a publication where the fertility of the whole world is declining. Sperm count is declining. So we thought, his proposition was that if it keeps on growing, one day will come, if you do this kind of trend analysis, the sperm count will be doomed. We thought, uska idea hai, mere ko bhi theek laga, but proof nahi tha. Within few years, and this is a real problem, right? And then within few years, some papers came where the whole world, you see from, if you read from there, Bolivia, Brazil, Chile, Denmark, India, Italy, Japan, United States, United Kingdom, everybody's sperm count is falling. It's an official publication. It's very dangerous. We will all become childless. So this needs to be controlled. And these are the publication's name. If somebody wants, you can take from Indrashis or me. So we have gone to the clinical condition now. That if a man goes, says, I am infertile, I cannot produce baby, doctor will give FSH and testosterone treatment. There are two conditions. Either he is unresponsive or he is responsive and makes a sperm. So 50% of them respond, 50% do not respond. We call it idiopathic male infertility. In spite of hormone, God knows why he is not producing sperm. If I give injections, even then not. So what we started doing, that there must be, we thought hormones are given to the testes. I'll show you this, what it is. But they are not producing sperm. So must be these cells are defective. They cannot see hormones or they cannot respond to hormone signaling. So basically we thought that it must be a signaling defect. So we all know, if you have studied zoology, that this is a Sertoli cell, which is the mother cell. So if you see a pomegranate, there are a lot of granules, they are germ cells. And the septum in between is Sertoli cell actually. This cell feeds all the germ cells to grow and become sperm. So this mother nursing cell has receptor for FSH and testosterone. And if FSH and testosterone is shown to this mother cell, she does not respond because she has gone crazy. She is psychologically ill, mentally not in order. She will not produce goodies for the babies, and babies will die or will not proliferate to become sperm. That is our hypothesis, right? We have to prove it. So in science, we first come to hypothesis, then we prove it. So this is a testis. If it was made of a glass, I had to make, because I always was confused about size and shape of testes. These are the seminiferous tubule like sugar canes. These are blood vessels. So if you take one sugar cane out, you will find this is Sertoli cell. These are germ cells growing. And in the center, there is a well, lumen, where sperm is released. So what happens? What I was talking about FSH and testosterone. So what was happening? Patient went to doctor. Doctor, though I know this is a diesel required for this FSH. And I know you require testosterone, that is the current. You will be giving from day after tomorrow sperm. I'll put some diesel, FSH, put you testosterone, current. And he thinks these germ cells will go here and make sperm. Ideally, they make sperm. But does that, gives hormones, no sperm comes out. So who is defective? This Sertoli cell, my mother cell, who is supposed to take this signal, translate it into its own goodies. Let us say kheer, chamcham, aloo bunda, gulab jamun, and give to germ cells to become sperm. Sertoli cell is not working, suffering from fever, not well. Genes are not functioning. So we dissected it, the pump, and tried to find out what kind of signal mechanisms are going on there. So basically, this is a Sertoli cell. So I always competed with my students. Either you make as good as I do, my imagination, or you will not survive. So they, I hope, still do that. So Sertoli cell is orchestrating all sorts of the germ cells, spermatogonia, spermatocyte, spermatid, and sperm, with different music at different point of time in a song like that, and making sperm. If Sertoli cell is not well, defective in genes, it will not make sperm. So what we are trying to say, there are genes helping getting all signals and making several mRNAs, making several proteins, they are going to germ cells and feeding. This is the normal situation. All signals are received by the Sertoli cell. So this is a mother cell. Koi bachche ko, she gets to play, somebody feeds, somebody calms down, somebody when grows up to gives to read. Mother has to decide which baby wants what. Spermatogenesis is a process from a small cell to tailed long sperm is made. Sertoli cell is the mother nursing cell. If she is not well, she has genetic defects. Doesn't matter how much hormones I give, how much FSH testosterone doctor gives, how much diesel and electronic energy or electric energy put in the machine, nothing will come out. Nothing will come out. So what's happening here? That probably I am giving all the hormonal signals to cell. The genes are there, but this gene was defective, this gene was defective, and this gene was defective. So mRNA was defective, everybody produced protein, these did not, and these did not. So, although everything was there, tel laya gaya, mitti laya, gas laya gaya, लौंग इलायची दारचीनी सब ला गया, पकाने वाले कुछ सेल्स के प्रोटीन्स थे, खाना नहीं बना सकते. Still you can't eat. Sperm will not be made. So we dissected out what are these things. I will go very fast from here because I have explained you everything. So in puberty, you have a lot of hormones, you make sperm. In infancy, you have hormones are there. Hormones are there in infancy, but we do not make goodies by the mRNAs, and so there is no spermatogenesis. I'll tell you, this is what is infancy. From 0 to 6 months, a boy is like his father in terms of hormone. Then God shuts off GnRH, main hormone impulse. Every hormone goes down, and then at puberty, God turns on the brain. Again, the hormones come up. But at that time, Sertoli cell was naive. You can see it's very small. It doesn't respond to hormones and doesn't make sperm. During puberty, when hormone comes up, it makes the sperm. It gives all the ingredients, all the genes are turned on, and it gives the sperm. Sir, how many minutes has happened? 10 minutes remaining. I'll finish. So whatever I can finish, enjoy that, right? So I have now turned into signaling, real sense signaling. So this is Indrashis Bhattacharya's work. So we have published series of paper dissecting the cell and looking into G-alpha coupled receptors, G-alpha S expression and all. I will not go in detail, but I can tell you the punchline. This infancy, hormones are high, cells are blind, cannot see all hormones. During juvenile period sometimes, there is a switch which occurs into the train by that signal, and the train turns to puberty, and you can make the sperm. Some individuals, this switching does not occur. Their receptors remain defective. So what happens? They keep on remaining pubertal for rest of life. So they do not basically produce sperm. It's a large amount of data. So what I'm going to say is this is the period of infancy, juvenile and puberty. There are a lot of published papers. This is a paper with Souvik, Indrashis and myself, which has really reviewed the whole scenario. Don't worry. I'll tell you some funny things. This is a germ cell sitting in the testis. I assume it is sleeping. Why? Because not until 13 years of age boy produces sperm. So germ cell is sleeping. I assume it's sleeping. But what happens? Now I have shown you so many things. If this is Sertoli cell, so somebody will say, sir, germ cell to thik thak hai, sir, daudta hai bachcha. Sertoli cell is sleeping. I don't know. So Sertoli cell is sleeping. Probably that is why germ cell is sleeping. Let us assume. I am giving FSH and testosterone to Sertoli cell. If it wakes up, boy will wake up and play and make sperm, become a big guy. Now, if that does not happen, that signal will not go and he will never wake up. So this is what happens in infertility. So if I try to catch testes of a man and say, let us take out some testes and let us do some experiments on your Sertoli cell, he will run away. Man will not. है ना? हाँ क्यों कर रहे? आदमी देगा, testes नहीं देगा. She is looking, So we have taken monkeys because human beings are like monkeys. Look at how smart. He is drinking Sprite in front of me. He snatched somebody's Sprite at a very cheap phone of 1600 rupees that time. इतना ही अच्छा फोटो आता था इससे नहीं. But you see, they are very smart. So we have taken monkey because they are non-human primates, similar to human. Did lot of things, hormone manipulations to take out their Sertoli cell and culture them, and again do the same thing. Consider that infant Sertoli cell is infertile because it is seeing hormones but not producing sperm, and there is a fertile testis. And in lot of studies of differential genomics, proteomics, transcriptomics, I will not go into those details, and published lot of paper in several big journals about these cell signaling mechanisms inside the testes. This is Indrashish's work, which shows that during infancy, those hormones are defective, and especially G-alpha-S, it's a published paper, was defective. And during period of time, when puberty, the G-alpha-S becomes very defective, and these goodies are very less brown colored, and you can see lot of goodies here, brown colored. And then they go into the nucleus, do transcription, and do a lot of good things, and we get the sperm. I'll stop there. I'll come to another work of Souvik. We did a new signaling mechanisms, first time published. So what is YAP? Yes-associated proteins. It's a co-transcription factor for the hippo pathway. Some of you, if not know, whenever you read. So it's crucial for the often oncogenic regulator of organ size, tissue regeneration, cell proliferation, and when activated, it moves from the cytoplasm to the nucleus to promote the gene expression. So this is hippo pathway is given there. So what we found, it's all written, but I will not go there, I'll just show you the data. What we found, that when we are looking at the various spermatogenesis related genes, we found that when we give forskolin, it should stimulate the cyclic AMP production, and so the spermatogenesis genes are upregulated. When we give vertiporphin, which suppresses YAP, if it draws YAP action, whatever increase because of cyclic AMP, when we give vertiporphin, which depresses YAP, it comes down, proving that YAP is regulating cyclic AMP somewhere. Same happened with everyone, and this is the data. I'll go there. Now phosphocrab, you know that it has to move to the nucleus for transcription. Do you know that? For transcription machinery, pCrab is essential. So when you give, this is untreated, when you give vertiporphin, YAP is suppressed, there is a lot of pCrab in the nucleus. Now protein kinase A is a molecule next to cyclic AMP which works, if you know signaling. If you give H89, which is suppressor of pKa, this nuclear localization is gone. Clearly indicating that YAP is one of the bosses sitting in the line of signal transaction and managing कि बेटा मैं सब signal transaction सही है, signal be waived, I am the police, I will come and stand here, I will say stop here, go there, don't go. So for the first time we have shown that YAP is a new molecule which works in the testis. It was never shown before. Now we have done a lot of differential genomic studies between infancy, less hormone, same hormone but no spermatogenesis, and puberty, hormone and lot of spermatogenesis. And what we did, we played very smart. Understand, in childhood there are hormone, no sperm. This gene is upregulated. We believe this gene is suppressing spermatogenesis. In adulthood, you will find this gene is low. So we believe this gene is suppressing something to allow spermatogenesis to occur. We did transgenesis in a different manner. Either we suppress this heightened genes in infertility by shRNA, live animals, by transgenesis, or we overexpressed that gene in adulthood and tried to see what is the result. So my job is basically to see differential genomics into real-time things. This is the DNA microarray work, like this. There are a lot of genes which are coming up, and our job is to crisscross and express childhood genes in adulthood, see if they become infertile. I'll go only with that. I'll not go to the other thing. So I've shown you this. This is, can you click inside? This is another way of making transgenesis. This is buffalo embryo, and you go, and this is what is cloning. This we have published paper. You go inside and remove this. This is a polar body, hex dye. You remove this genetic material and throw out. So you have now an egg without any chromosome. Then we insert transgenic chromosome in that. So these genes are transfected with genes of interest, and these cells, and chromosome or nucleus from that will be injected here in this enucleated egg, and the baby born should be green because I use green fluorescence protein. And luckily, what we found that out of the 2, 1, and out of the 3, 1 embryo turned green. So these are the way you can do transgenesis. These are not easy ways. You need a very big equipment. You are killing hundreds of mice to make some transgenic animals. So the methods of green is DNA sequences and genomics went up, but the blue technologies of transgenesis remain low. So we did another innovative work. What to do? If we do egg-related transgenesis, 6 months में एक transgenic animal होता है. And now we know about so many genes. I want 10 transgenic animals per month. How do I do that? So we discovered a new technique. We found that this crossing over occurs at the time of cell division. Let us imagine a lady has come to dance here, very famous lady. It's an organized dance. There's police commissioner and governor, everyone is sitting, and a man gets excited, says, I want to dance with her. Police will come, catch him and take away. You can't dance. This is organized dance. 30 days ago, we advertised, she's a very famous lady, you can't. The man gets frustrated, he's released on bail. In the evening, he goes here at disco. There also he sits for 10 minutes, gets very excited, I want to dance, he joins. Everybody says, come on, dance. Doesn't matter. Why? Because cell division is in haywire. Nobody knows who is doing what. That is what exactly happens during embryogenesis. In 21 days, whole animal is to be made for mice. So what we did, we thought, similarly in testes, a man produces 200 million sperm in a day. Huge amount of mitosis, meiosis, bhangra, dance, insert gene there. Rather than egg, you have to kill animals, superovulate animals, 100 animals to get so many eggs, inject. Who will do that hard work? So we started doing this work, and I'll again go into short, maybe that's possible. Let's try. And what we did in testes, we injected GNA. We did an electroporetic current, so the germ cells opens mouth, DNA goes in, and we could produce transgenic animals, and it's a wonderful work. This was published in Nature Methods. That worked. What we thought worked. So everybody wants happiness, nobody wants pain, but you can't have a rainbow without little rain, so work very hard. You will get the results. You see, look at this man, be bold. He's walking on Delhi, half pant, he's wearing tie also. I didn't dare to go in front and take photo. He goes his own way. He thinks right. What he's doing is correct. You should think that way. Be free. Don't think कोई बोलेगा, हंसेगा, testis में जीन डालना है, current मारना है. क्या बात कर रहे हो sir? No, don't do it. It worked for us. Something else will work for you. So now I will go to various genes. So we did this comparison. And my job is DCOP-K3, one of the genes I have taken, which is highly expressed in puberty, and it is associated with spermatogenesis. I'll try to shut off DCOP-K3 during adulthood and try to see, do I get infertility like an infant? Because in infant, DCOP-K3 is less. So high pubertal DCOP-K3, we used SH, this again we proved by real-time PCR in monkey and mice. Yes, DCOP-K3 is high brown pubertal as compared to infant violet. So when we made a construct, I will not go into that. We successfully declined the mRNA in the Sertoli cell, mother cell, because we used promoter of ROX5, which is promoter of Sertoli cell only. We haven't affected other part of the body, only Sertoli cell. mRNA of DCOP-K3 went down. You can see Western blot, you can see the colors. DCOP-K3 went down. Now you have to say, does it cause infertility? Sperm count went down. Litter size in F1 generation become almost half. So if I mate mother-father now, it will become homologous and you can get the infertility. This is the condition of the testis, very bad on the right-hand side. And what is that? Is basically DCOP-K3 is known to suppress Wnt signaling. As soon as you shut off using shRNA mice, this DCOP-K3 shuts off, Wnt signaling you can see. As soon as you shut off DCOP-K3, Wnt signaling is on. As soon as it's on, it retrieves everything, every signal molecule to its own, and beta-catenin will be not phosphorylated, and it will go sit in the nucleus and do the signal transduction, that is keeping the germ cells like an infant. Let us see what happens, and this is how the fertility is regulated or down-regulated. I have 5 more minutes. Thank you, so nice of you. So I'll say another, this is microRNA. So I have shown you DCOP-K3, it's a protein. We have picked up microRNAs. This is one of the microRNAs I will concentrate, MIR382P, and we generated a mice of MIR382P, and MIR382P is actually, if you go to transgenic mice, where MIR328P is small or low in puberty. So if I overexpress, I must get infertility. So what we have done, we have overexpressed in mice, and this is the body weight, remained good. This is the testicular weight, decline. This is gonadosomatic index, decline. Testosterone levels more or less remain non-significant. And you can see the testis has become small like an infertile guy, and this is a big testis of a normal guy, successfully. So we have seen a protein, we have seen a transcription factor, we have seen a miRNA, and Kamal Mandal has done on transcription factor, and there also we found testis jalebi kaise dikhta hai. Dikha nahi raha hu. We have taken another 382-3P, which is, this is what is the sperm count decline. All the males were infertile as compared to wild type. The single gene, such a nice effect, wonderful thing. Similarly, we have gone to MIR92-3AP. It has also the same signaling cascades, which in childhood keeps the Sertoli cell as a small Sertoli cell. As soon as the FSH comes, it suppresses MIR35. These suppression on good genes are gone. So what we have done in adulthood, we have chose to, I'll take 5 more minutes to finish, right? In the adulthood, differential expression analysis of qPCR we have done, and what we found that if we try to manipulate expression of 92-3P in adulthood, that is overexpress that, it will overcome the FSH-driven signal transduction, and it will suppress all of them. And this Sertoli cell, which is supposed to become a big Sertoli cell in adulthood and make a lot of sperm, stays like that. And we did that experiment. It worked exactly what we felt, and that was a beautiful story. So I'll tell you what is signal transduction, what is physics, what is chemistry. So gravitation cannot be held responsible people falling in love. Gravitation cannot be held responsible for people falling in love. How on this earth can you explain in terms of chemistry, physics, and I would say cell signaling in the important, in terms of chemistry, physics, as important as the biological phenomenon as the first love. What do you need? You need cell signaling. Only chemistry, physics will not do. What is that cell signaling? Put your hand on a stove for a minute and it seems like an hour. Sit with that special girl for an hour and it seems like a minute. वो burning पे रखोगे तो acetyl choline पता नहीं क्या क्या, you just take out the hand. But this is what is biology, and this is so important to dissect biology in a good manner. This is my last work, and I will finish this. Lot of drugs are now made which are to be replaced in human because they cannot make it because of cancer or some other disease. So we do make a lot of drugs, but they are very, very costly. These are the diseases, arthritis, and this is Guillain-Barré syndrome. But if you replace, many of them become good. Cost of them is exorbitant, 40,000, 7,500, 15,000, one dose. Why? Because this is made in laboratory. You need large fermenters for bacteria to express those human genes, land, manpower, 3-shift manpower, electricity, maintenance, everything. But if we can take the promoter, that engine of a protein which is produced in udder gland, and remove that pipe and put human gene and make a transgenic animal, possibly in the milk, it will make the human protein. That will be cheaper. Then this is your industry eating 450, 500 rupees per day, and it will secrete milk along with the human genes. So this is called as biofarming, and this we have successfully done. We have isolated promoter of buffalo beta casein, that is a promoter of the udder gland, tagged human gamma interferon to that, don't go into all these details, and EGFP. And after birth, we found udder gland, only udder gland is green. This is the whole dissection, but only udder gland is green. And when we killed the animal, we found neither spleen, liver or kidney, heart or brain, but only mammary gland expresses GFP protein. indicating it is very specific and it will be produced only in the milk. Then we have made transgenic animals, taken out the milk, and we have, remember, we have taken the human gamma interferon kit from the market, human gamma interferon. And in transgenic animals milk, we can find this much amount of human gamma interferon as compared to controls. It's a very good work. It is also a cell-based work. I was talking about cell technology. Let us pray to the God that we are successful in transfecting mammary epithelial cells and make such kind of human proteins. So some 14,000, if we can make rupees 200 or 300 vial. This is all a wild guess. And I'll be suggesting, this is my last slide, maybe last but one, if time is there, that all of you should dream very high. We have done a lot of innovations using cell technology. We have dissected the whole testicular cell signaling, and around 10 genes of cell signaling in testes we have discovered which were not known before, which are related with those untreated human male infertility. And since we work with animals, and I would suggest all of you that please take care of animals while you work with them. They deserve and expect love from everybody on this earth. Thank you. Thank you, sir. That's what you have to learn. 40 minutes means 40 minutes.
Thank you very much for a very good presentation that bridged the interest of students and that of scientists that are longer in the field. So well done. It's open for discussion. Are there questions? So there's a question over there.
Okay. TADA form ka to nahi hai na?
Sir, are there any established strategies for the recycling or the recovery of the exploited cells?
Can you again tell me?
So, are there any established strategies for the recovery or recycling of the exploited cells? When cells are exploited, can we recover them or recycle them?
Yeah. It's the beauty of it. When you are inserting your genes of interest, insert genes of AGAP, insert genes which are expressed on surface so that you can run it through a column and also get tagged. We have done those kind of work for sperm. So when we do testicular transgenesis, in mice we put for mating, 12 babies are born, 2-3 are transgenic. If I do it for a cow, 10.5 months later I get a baby. It may not be transgenic. So what we do now, along with this is a bicystonic construct. So there is a gene, for example, beta-casein promoter, human gamma interferon gene. I want that to be produced in a baby in the milk. But when I give testicular injection for transgenesis, out of millions of sperm, maybe 200 are transgenic because everyone has not become. So what we have done, along with that gene, we have tagged a germ cell specific promoter, let us say SP10. Under that, we have tagged EGFP, that too extracellular localization signal. And then you do FACS. All green sperm, 200 are sorted out. You use that for IVF and embryo transfer. You can do that. You have to use the double construct.
Are there more questions? If not, then I have a question to you. So you mentioned a transcriptomic analysis on pubertal and post-pubertal testes in monkeys. And you said that in terms of the gene ontology, there were a number of pathways upregulated in the prepubertal testes. So can you flag which ones these are that then are upregulated?
We have many of them. If you want, I can give you the references. You can study them. And we have really done real-time PCR also. So you can pick up the best ones. We have worked with 1 or 2. Rest of them are there. If you want to work, there's a number of them. Yeah. So I have shown 92 3PA and MER 382P or RODA, it's a transcription factor, or DICOF K3, which is a protein. Like that, we have several molecules. I can give you reference. Thank you.
Sir, one question, can I?
Sure, madam.
Yeah. Sir, really appreciated for your wonderful presentation.
Thank you.
And definitely all of us, not only the students, but all, we enjoyed very much.
Thank you.
I have one question, sir. You said that the hundreds of mice are killed just for your experiment.
Yes, of course.
Okay, this is not exactly related to your research because you only need the testes, right? So in order to take the testes out, is it possible to the survival of the mice again? It could be possible, what I understand.
Yeah,
Why
yeah.
you are killing the...
No, what I am doing, I don't even touch the mice. I just give electric current. His testes has genes, which has foreign genes. I put it for normal mating. That male doesn't die. I don't do any surgery. But for the oocyte mediated, you have to kill the mice, cut open the abdomen and remove the oviduct after the ovary, where 30 eggs come if I give hormones. And you have to manipulate them in vitro, as I have shown in video.
But hysterectomy or that testictomy is not possible without killing
For
the animal.
for this experiment, if you want to do it egg way, you have to kill the animal.
Oh.
Yeah, because usually what happens, this process has to be very fast.
Okay.
Because you have to keep the embryos surviving or eggs surviving. So they don't care about the mice. They are more careful because every transgenic animal is very, very precious. Several diseases at today's date are understood because of these transgenic animals. I simply said a gene which you insert in this and mice becomes bald. That's the gene. We will be billion years, million years, I don't know what. That gene, if you can put somewhere in head, hairs will come.
Okay.
So that's how the field is. Nothing. That's I said. There are two ways. In testicular way, she is asking, do you cut the testes and keep the animals alive? I said, I do not need to cut the animal. In egg way, always people kill animal. So this is, our work is also more to ethically also superior. So our several places our title is ethically superior, human way of making transgenic mice. Thank you very much.
Yeah. Thank you.
Thank you, sir.
That was very nice.
Thank you, sir, for such an insightful talk and a wonderful presentation. I now request the chair and co-chair to present the memento as a token of appreciation to Dr. Subeer S. Majumdar.
So after a long, I took a class like class.